So it can't have escaped anybody's notice that there's been a revolution in our ability to measure nucleotides, DNA, the human genome, RNA, the message. But increasingly we're realizing that we have to take account of where in the cells those messages are. And so to tell us a bit more about that in the context of the Human Performance Alliance, it's my pleasure to introduce Sam Ward. Let me make sure I get his titles right. Professor in the Departments of Orthopaedic Surgery, Radiology and Bioengineering at UC San Diego. He also leads the Innovation Hub at UCSD and the Triton Center for Performance and Injury Science. Yeah, welcome.
Thanks. How's the mic? Okay. Thanks very much for the opportunity to speak. I'm excited to present a little bit of a unique perspective. I'm a clinician at heart, and you sprinkle a little engineer and a little bit of biology and physiology on that, and I think that motivates my questions. So like everybody else, I'd like to acknowledge my co-authors. Severin Ross is in the back. He is a postdoc who's worked hard on this project. And Adam Engler is around here somewhere. I'm not sure where he is, but he has been a faculty colleague of mine for a number of years. Rotator cuff disease is a big clinical problem, and it's a difficult problem to manage. It affects both the general population and athletes. And the manifestation at the level of the muscle is muscle retraction, fatty atrophy, degeneration, and fibrosis. And people love to look at these MRI images like you can see here. This region is supposed to be gray and full of nice, plump, healthy muscle. And you can see in this case, it's been replaced by fat and connective tissue. In fact, all of the rotator cuff muscles have been replaced because this is a massive tear. It turns out these are insensitive to rehabilitation, which is disappointing for the physical therapist in me. It's difficult to repair surgically, which is disappointing for the surgeons, and it's prognostic of high re-tear rates, which is bad for everyone. So surgeons had been noticing for years that it took more force to pull those torn muscle tendon units back out to their anatomical footprint on the humerus. And Christian Gerber sort of quantified that with a novel set of intraoperative instruments where he could measure how much force it took to pull back out and see if that was predictive of failure or a bunch of other physiologic variables. And he basically just produced a case series in the late 90s. These were all patients with chronic tears versus somebody with a very acute tear. And you can see that the people with chronic tears take more load to pull back out to the humerus, suggesting the muscle's stiffer. Pretty straightforward. About 20 years later, we decided to get a little fancier and went in intraoperatively and used a pituitary runger to grab a piece of muscle, take it back to the lab, and do mechanical experiments on single cells, or bundles of cells packaged about the size of a fascicle. And the data basically shows at the top here that single cells are no stiffer after a tear, but bundles of cells are significantly stiffer. And this was our first indication maybe we should be focused on the matrix and not on the muscle cell itself. Adam and I had a PhD, had a PhD student who was interested in this problem, and she used mass spec proteomics to characterize the matrix in people with tears, big tears on the top here, and no tears on the bottom. And what she showed was qualitatively and quantitatively, there's massive dysregulation of the protein structure in the matrix. But even more importantly, when she used that matrix from humans to challenge what they did in terms of cell behavior for progenitor cells in the matrix and near the matrix, it altered their cell behavior. So now you have a mechanical effect and a biological effect from the matrix. So these are interesting problems, interesting experiments in humans, but you just don't have the fidelity in time and space and biology to answer the questions the way you want to answer them in humans. So we spent a lot of time developing animal models. We've gone mouse to sheep and everything in between. In our hands, the rabbit model produces a histotype or a phenotype of the tissue that most closely resembles humans in a sort of a reasonable way. So this top row of images are different time points after tear in a supraspinatus muscle in a rabbit. You can see that the fibers are shrinking and there is space forming between fibers, which allows fat and connective tissue to accumulate, in contrast to the sham micrographs on the bottom. We can, of course, quantify that in a variety of ways, but at the bottom of the slide, you show atrophy very clearly, and you show progressive accumulation of connective tissue and fibrosis, which is exactly what we want to see for the model system for cuff tear. You can also repair it. You can repair it using the same tools that you would use to repair a human cuff. And again, if you look at the quantitative data at the bottom, let's see if I can get this to work, you see a classic finding in rotator cuff, which is a second hit of the surgery. You get further atrophy after the initial repair, and then it recovers just to pre-surgery levels in about 8 weeks, but never gets back to normal. And you get further exacerbation of connective tissue content in that muscle. Probably easiest to see here, all these small pink lines between the larger pink cells are fibrosis in that muscle, in contrast to the control muscle below it. Okay, that's fine, but the natural question that would come from here is why? How does that happen? How is it regulated? So we had really great time series data, we had great histology and great physiology on those muscles, and we took a hunk of that muscle, ground it up, and sent it off to the sequencing core like everybody else does these days to look at transcriptional profiling. And these are just volcano plots that come back. They don't tell you the story about what's going on transcriptionally. They just tell you maybe the volume of transcriptional dysregulation. You can see at 1 week, for example, all those blue dots are either significantly upregulated genes on that side or significantly downregulated genes on the other side. It doesn't tell you what they do, just that there's a big transcriptional response. Peters out in about 8 weeks, and then reemerges at 16 weeks as a different story. So if we assemble the connection between those genes and tell you the story, just in summary, there's a strong initial atrophy in immunogenic response in that muscle to tear, and late, there's a resurgence of a different immunogenic activity and a fibrotic response that results in the fibrosis late in those muscles. Interesting, but now what happens when you repair it? Similar story, lots of transcriptional activity early that peters out over 8 weeks, but the take-home messages are, if you repair that tendon and you allow the animal to reload, you get no hypertrophy or regenerative response in that muscle from the whole muscle transcriptomic perspective. Which is not surprising clinically because of what we see in humans, but it is surprising from a biological and physiological perspective because if the muscle is reloaded, there's no reason why you shouldn't have an anabolic response. In fact, you see exacerbation of inflammation, lipid metabolism and deposition, phagocytosis, apoptosis, other biological jargon words that mean that the muscle is generally not happy with what you've done with it. That's interesting, but a complicating part of that transcriptomic story from a whole block of muscle is I just got done telling you that the proportions of that muscle are changing over time. You have more fibrosis in it late. So spatial transcriptomics might give us some insight into what's going on more regionally in the muscle. And Severin implemented this work with 10x platform. I'm assuming it's the same one that Keith used earlier. He's nodding yes. So essentially the way this works is you take a tissue section, just like we would normally cut for fiber size, lay it on a special slide with 5,000 dots on it. Each dot's about 50 microns in diameter. It has the whole transcriptome on it, and it has a barcode. So after you do the preparatory steps, grind up the tissue and send it back for sequencing, all the transcripts are then geolocated to a spot on the histology slide to allow you to look more in a more granular way. So in this proof of concept paper that Severin wrote, if you just look at unbiased clustering, you see transcripts that coalesce around certain transcripts. And if you take that unbiased clustering and lay it back on the top of the muscle, it sort of makes sense. The green areas which are blown up here are rich in collagen isoforms and fibronectin. They fall in perifascicular planes in fibrotic areas. The blue and orange dots are rich for myosin isoforms, which are consistent with what you should see in a muscle. So generally speaking, it seems to work. If we then take those tools and apply them to the exact same animals that we had histology, physiology, and bulk transcriptomics data on, you get principal component analyses and a whole array of analyses back. The right way to think about this is the colors represent different groups like normal versus tenotomy, and the more separation between colors, the more different the transcriptional responses are. Similarly, you can look in more detail and look, for example, you could compare the transcriptional activity of a connective tissue compartment between normals and those that are repaired. So it gives you more granular detail. And I'll just tell you one part of that story that I think is pretty interesting, and I'm going to simplify it and just show you sham control is the top row. This is what the histology looks like. 8 weeks of repair and 8 weeks of tear. 8 weeks of tear plus 8 weeks of repair. The dots and the colors on the dots represent transcriptional activity for the given gene that's listed at the top. So for example, collagen 1 was upregulated in the bulk transcriptomics data, so no surprise, but is that collagen expressed ubiquitously throughout the muscle, or is it concentrated in areas of fibrosis or connective tissue planes? The answer was it's expressed more ubiquitously in the muscle after tear, and you certainly see concentration of it in these fibrotic planes after repair. Probably more interesting were there were genes that emerged from the spatial transcriptomics data that weren't even on our radar from the bulk transcriptomics story. So this gene MGP is just one gene. It's a more complicated story than that, but one gene that's pretty interesting. And that gene regulates calcification of fibrotic activity, and it's also permissive of muscle regeneration by inhibiting myostatin activity. So that gene never showed up on our bulk transcriptomics data, and it's highly expressed in regions of fibrosis after repair. Probably most important was the bulk transcriptomics story said there is no hypertrophy or regenerative activity in the muscle. And if you look at neonatal myosin heavy chain or MyoD, two transcripts involved in the regenerative process, they are expressed after tear, and you do see them in pockets of muscle after repair, suggesting that there's some regional potential in that muscle. So to summarize, spatial transcriptomics yield novel insights relative to bulk transcriptomics. That's an important lesson for us. Repair, and I gave you two examples. Repaired muscle has some residual regenerative capacity, which we did not see before. And there are novel genes in the muscle matrix interactions that we couldn't see in bulk transcriptomics. Both of these things could be targetable with appropriate timing. And then more broadly, as it relates to performance, tissues, musculoskeletal tissues in particular, have regional variability in loading. And these biological tools may allow us to capture some of those unique biological responses to differences in load. And then, of course, my goal as a clinician, optimized recovery from muscle or tendon injuries may require manipulation of this muscle matrix interaction to restore function, because we don't accept the idea that after injury with conservative care or surgical repair, you wind up with this sort of chronic decrement in function of a muscle tendon unit, and therefore the joint, and therefore the person. We can do better than that. Thank you very much.